Alright guys, welcome to chapter 7 of Concepts of Biology. Today we're going to be going over kind of the cellular basis of inheritance, meiosis, and a little bit of kind of gene swapping that goes on. So like us, most large multicellular organisms will begin their life as a haploid egg that then gets fertilized by a haploid sperm cell and then will grow into a diploid multicellular organism. This happens in hippos, this happens in flamingos, and it happens a little bit as well in plants. So it is a multi-step, evolutionarily adept process. So in part 1 here, we are going to explain the variation among offspring and how that's an evolutionary advantage that we get from sexual reproduction. And we're also going to talk about the 3 different life cycle strategies that multicellular organisms use for sexual reproduction. We talked a little bit about sexual versus asexual reproduction. Sexual reproduction requires a partner of the opposite sex. Okay. Sexual reproduction requires a partner of the opposite sex of the same species or a species that is similar enough to reproduce with. Asexual reproduction is a solo act. Essentially, that organism will have the exact same as their parent. While in sexual reproduction, you get a unique offspring every time. Okay. Some of the pros, there are pros and cons to both. Some of the pros of sexual reproduction, you get genetic variation, you get possible evolutionary advantage, all kinds of stuff. Asexual, some of the pros of that are less energy trying to find a mate. And if parent is well suited for the environment, offspring is be a-okay. Good too. Okay, that was long. Some of the cons of asexual reproduction, you're not going to get no variation unless mutation. Okay. Like this just says what causes variation in the offspring. For sexual reproduction, it is meiosis. And for asexual, it's mutations. Okay. So, some important terms that you need to know. Diploid. Diploid just means a cell that contains 2 complete sets of chromosomes. And you get one of those complete sets from each parent. A somatic cell is a non-reproductive cell. So that's going to be like your body cells, your muscle cells, your epithelial cells, any type of cell that is not a reproductive cell. Haploid means a cell that contains a single set of chromosomes in human, that is an egg or a sperm cell, which is also known as a gamete. A gamete is a sex cell. Okay. A germ cell is a special cell line that only makes gametes. When we start developing in utero, we start forming from three main different types of cells, but that's an embryology problem that you don't need to worry about right now. Just know that germ cells are a special cell line that only makes gametes. Meiosis is the process of cell division where a cell goes from being diploid to haploid, which is very similar to mitosis, which just copies the entire genome. Okay, and then fertilization is when two haploid cells form back together and make a diploid cell, and that's where all of us came from. Okay. So in this diagram, you can kind of see it's a little blurry, but it's basically just showing you that in animals, adults are formed from the haploid gametes. The haploid gametes being the egg from the female, the sperm from the male, that will eventually form together and make a whole diploid offspring. Okay. Fungi, anything in the mushroom kind of family category, will have a haploid dominant life cycle. So for most of its life, it will be haploid, and then while it is reproducing, it will be diploid. And then plants can go either way. Some plants will be haploid in nature, others will be diploid in nature. It just kind of depends on the plant. This is just a long paragraph that says basically that sexual reproduction is a way to form a unique diploid cell. I'll highlight the things that you really need from this. Sexual reproduction is the product of a haploid cell and the fusion of a haploid cell to form a single unique diploid cell. This diploid, that doesn't need to be there. Diploid cell will undergo mitotic cell divisions to develop into an adult organism. Meiosis and fertilization introduces variation into offspring that may account for evolutionary success of sexual reproduction. And then the vast majority of eukaryotic organisms employ some form of meiosis or fertilization to reproduce. Now that we've introduced that topic, let's talk about meiosis. So in this section, we will discuss the behavior of chromosomes during meiosis, how they act, where they go, what they may be doing. We're going to talk about the cellular events that occur during meiosis. We're going to explain some of the differences between meiosis and mitosis, even though they are very similar. And then we're going to explain some of the mechanisms within meiosis that create some genetic variation in the offspring, or the products of meiosis, which are the offspring. Okay. So if you remember from mitosis being just regular cell division, there's kind of 5 phases that we talk about, and then an unofficial 6th. And that's interphase, prophase, prometaphase, metaphase, anaphase, and telophase. And that unofficial sixth is cytokinesis. Okay. In meiosis is very similar, but it has some major differences. One of the major differences is that meiosis actually occurs twice. It is two cell divisions. We go from having diploid cells have two, and it n equals like complete number of chromosomes. So we go from going from diploid cells to haploid cells. Haploid cells having half. Okay. The end result of meiosis is 4 haploid cells that are different from each other and from the parent cells. Okay. So no two eggs are exactly the same. A question that might pop up when I say that is twins. Twins happens because the egg splits after it's been fertilized. So it's the same egg. That's at least monozygotic twins. The same egg that got split twice. That'll be that got split and formed into two different beings. That would be monozygotic twins. And those are actually pretty rare, identical twins. Okay. So meiosis reduces the number of chromosome sets from 2 chromosome sets to 1, and that genetic information is mixed during the division of meiosis, which makes some unique chromosomes. That's why no two eggs are the same, no two people are the same. Even if you have siblings, you and your siblings are not carbon copies of one another, nor are you carbon copies of your parent. That is due to this crossing over that occurs in meiosis. There's meiosis 1 and meiosis 2. Meiosis 1 is basically just mitosis. And then meiosis 2 is where everything kind of splits a little bit differently. Okay. But in meiosis 1, prophase 1, the homologous chromosomes, so the ones that you got from mom, let's not do this highlighter, sorry. The ones that you got from mom, your complete set of chromosomes that you got from mom, and the complete set of chromosomes that you got from dad get together and form a tetrad. So these guys are hanging out together. They're besties. If I could draw them again closer to each other, I would, but that's what happens. And as they are close to each other, you will sometimes get a little bit of crossing over. So the genes that would be in this bit of the chromosome and the genes that would be in this bit of the chromosome kind of swap places. And then you get a little bit of genetic variation. And it's kind of like that. So they just kind of swap. So the same genes will crisscross, so you don't lose any genetic information. It is just different genetic information. This is what that process looks like. We start with interphase. We do all the growing, the synthesizing, the duplicating of our genome. Prophase 1, our homologous chromosomes join up together. Metaphase 1, we separate those homologous chromosomes. We go through the rest of the mitosis bit. We cytokinesis. We have 2 different daughter cells that are still at 2n of their genetic information. Okay. At this point, they then go through meiosis 2, and after metaphase 2 getting into anaphase 2 is when we have just one copy of the DNA on either side of the cell. Okay, so that's kind of how that one works. This kind of illustrates that chromosome crossing over that I was talking about. When your homologous chromosomes are next to each other, they're lined up, sometimes they will cross over and you'll get some recombinant chromosomes. That's what we call them. Recombinant chromosomes have a mixture of the maternal and paternal sequence. So they're a little special. Okay. So in prometaphase 1, the microtubules will attach to the kinetochores of the homologous chromosomes. Those are these guys in the middle that hold the chromosomes together. This is very, very similar to mitosis, and they will get pulled apart. Not apart from their sister chromatid, but apart from their sister chromosome, their homologous pair chromosome. Okay? And then in prometaphase 2, those microtubules will attach to the individual sister chromatids. So that's this guy, chromatid. Chromatid versus a chromosome. Okay? And this is the result of that random independent assortment. Sometimes you get some genes that will sort differently, and that's what allows for that variation. So independent assortment is essentially just the random alignment of those homologous chromosomes during metaphase 1. It could be any combination of that chromosome, but it has to be the same homologous chromosome, if that makes sense. So chromosome 1 couldn't join up with chromosome like 5 because they're not the same size. They don't have the same centromeres. They don't have all the things that could line up the same. So that independent assortment is random, but only to the extent of being the same type and size of chromosome. Okay. This chart is just kind of showing you the outcomes and the different things that happen in meiosis and mitosis and how that affects the amount of DNA that gets produced and where it ends up. There's lots of different ways in which meiosis creates new genetic combinations. And to summarize them, essentially, in meiosis 1, the maternal and paternal genes are recombined during that crossover that happens when the homologous pairs are together. And then the random assortment of tetrads at metaphase produces different combinations of maternal and paternal chromosomes. Okay. Remember, those tetrads is the group of mom genes, dad genes pulled together. You've seen this image. Oh, this is the same thing twice. Ignore that. Okay. Some variations that you can get in meiosis, as much as, again, this is a well-documented process, it is not perfect. Our bodies are not perfect machines, but they do a pretty good job. So in this section, we will explain how non-disjunction leads to differences in chromosome number, and we'll also talk about how errors in chromosome structure can show up through inversion and translocation. Okay. In the mitosis presentation, I believe you guys saw a karyogram, a karyotype. This is, again, one of those karyograms. Okay, so you can see that all of the chromosomes generally have a matching pair that looks very, very similar, that functions very similar, that exists in a similar space. Okay, that even includes the two X chromosomes. These are both sex chromosomes. And you know that this is biologically a female because there are two X chromosomes. There isn't an X and a Y. But sometimes what can happen during this meiosis process is that each chromosome, as each chromosome is lining up, sometimes they don't fully pull apart properly. Okay, so they don't separate properly. And because they didn't separate properly, one cell will have an extra chromosome and one cell will not have enough. Okay, that's what that non-disjunction is referring to in meiosis 1 and in meiosis 2. Because again, still, sometimes you will get that the chromosomes do not, or the sister chromatids do not fully separate from one another. So this one would be missing some genetic information, as would, and this one has too much. Okay. So some of the terms that we use when referring to individuals with different numbers of chromosomes, or referring to different changes in chromosome numbers. So euploid is an individual with the appropriate, quote unquote, appropriate number of chromosomes. This just means that they have 23 pairs, as well, in humans. In humans, they have 23 pairs. They've got 46 chromosomes total. Okay. It's 22 pairs of autosomes, so, or body chromosomes, and one pair of sex chromosomes, making for a total of 23. Okay. Aneuploidy is an individual that has an error in the number of chromosomes they have. You can either have monosomy, in which you have lost a chromosome, or trisomy, in which you have gained some chromosomes. Okay. A common one that we hear about is trisomy 18, which is Down syndrome. Okay. A lot of the time, aneuploidy results in a miscarriage. There are some that do not, such as Down syndrome, trisomy 18, but aneuploidy usually, unless it's one of these specific chromosomes that are non-fatal to have not the right amount of, will result in a miscarriage. So some of the smaller chromosomes, 13, 15, 18, 20. I actually think Down syndrome is trisomy 21. Sorry. 21 or 22, that can result in an entire life being lived, or survival that only lasts for a couple of weeks here and there. It just kind of depends on which chromosome it is and how much of it is missing. We find that as maternal age increases, the percent likelihood of having eggs with aneuploidy rises. Okay. How is it that females have 2 copies of X chromosome? It's because of X inactivation. X inactivation is essentially a process that will use the genetic information from one of the X's, and anything that isn't needed will get turned off on the other X. Okay. And that's what results in a tortoiseshell in cats. You very rarely will see an orange female cat, generally because that second X chromosome will often get inactivated. There are some sex chromosomal abnormalities that we know about and are aware of. So 2X chromosomes, the embryo will become genetically female. And an X and a Y, that embryo will be genetically male. This has nothing to do with a person's gender expression. It has nothing to do with how they view themselves. It is just the biology of an X or a Y. Sometimes you will get a person that has 3X chromosomes. Oftentimes, a person with 3X chromosomes will be genetically female. And they will have a lot of X inactivation, sorry. Okay. XXY forms what we call Klinefelter syndrome. And in Klinefelter syndrome, generally, people will present as male, but they will have a total of 47 chromosomes instead of the normal 46, and they might have a few issues with kind of underdeveloped testes. They might be a little bit taller. They might have a few learning disabilities here and there, and the same thing with the triple X syndrome. Okay. If someone only has one X, they will generally present as female, but it can occur in males as well. It is a relatively rare genetic condition, but it does cause like a range of symptoms. Can be like being relatively short, delaying puberty, some ovarian issues, other things like that. And we call that Turner syndrome. Chromosomal abnormalities are usually found by cytologists. Cytologists just study the cell and a bunch of the different structural arrangements that can happen during meiosis. So we can get partial duplication of the genome, you can get deletions of part of the genome, you can get inversions of different nucleotides here and there, and you can have translocation, and that's just where a piece of the genome goes where it's not supposed to go in that crossing over or random assortment thing that we talked about a little bit earlier. Okay. Which can result in a host of genetic disorders such as Cri-du-Chat syndrome, and this young gentleman has that. Okay. So inversion is where a piece of that chromosome segment breaks and then flips, so it's in the wrong order. Reciprocal translocation is when two homologous chromosomes switch, but non-homologous chromosomes switch, but they don't lose or gain any genetic information. Okay, so this one's showing that reciprocal translocation. They moved to the wrong spot, and they flipped.